Thank you. Good afternoon. So from reductionist to public health, it's a quick transition. I received some of my research support from the private sector, and I serve on scientific advisory boards of pharma, food, and supplement companies, at least a few of them. So I thought I was going to see this slide a lot more often today. But for vitamins, essential minerals, and fatty acids, this is a pretty good model. It works pretty well. But what happens when we talk about other kinds of other constituents in our foods and in the food supply? We do have terms like acceptable daily intakes for food additives, and presumably there's no EAR, there's no RDA for them, but the acceptable daily intakes are presumably safe, and there is at least some safety evidence that would support it. When we talk about food contaminants, at least in the EU, there are tolerable daily intakes or tolerable weekly or monthly intakes. Again, we just ignore the left hand of the curve. We don't have to worry about essentiality in those. I think one of the challenging issues now is how do we look at phytochemicals and zoochemicals, all these non-essential compounds, the polyphenols, the carotenoids, phytosterols, alkyl resorcinols, where evidence suggests there is some real benefit. We don't have deficiency states for these compounds, although I would argue that there are probably a few where if you went through a full DRI process, you could actually identify lower levels of intake or absence of intake that do result in inadequate function or increased risk for chronic disease. I'll give you my bottom line without an illustration of where I'm going, and that's that I think when we start talking or contrasting this topic with traditional toxicology, I don't think we can talk about harm in nutrition without balancing that with benefit. This slide shows readily that there's harm, potential for risk with too much and for too little, the deficiency syndromes, but I think we can certainly see benefit with non-essential compounds and the potential for harm as well should the dose be too high. Well, how do we deal with that? But before I go on a bit, I just want to talk about this sequence of indicators for hazard and harm. So we have an acceptable daily oral intake range that WHO has talked about, but when we talk about biomarkers, and we've heard a bit about that before, particularly biomarkers of harm or toxicity, what biomarkers are we going to use? Are we going to use biochemical markers that actually don't have any significant pathological or clinical effects? Do we look at subclinical biomarkers, some sort of functional deficit? Do we use as the range of upper or lower intakes where we begin to see actual functional effects? I do think, however, we wouldn't disagree that death might be going too far before we decide to pick a cut point. But these are very important issues. I think the calls for biomarkers are well recognized, but we have so few that are considered to be validated, we can't use a great deal of the ones that I think are good candidates to start considering now. There's also the other issue as we look at this range of acceptable daily oral intake. I could make it a little broader if I used one of these lines for deficiency and another for toxicity. So how cautious do we want to be in using a biochemical indicator or a subclinical biomarker of deficiency versus one for toxicity? There is no rule that says you have to use the same on both sides of this U-shaped dose-response curve. I think another concept that's really important to keep in mind is that as we look at curves like this for EARs and RDAs, that these criteria are dependent upon, or these values are dependent upon the criteria selected for adequacy. And this slide would suggest that we could use different parameters, and indeed that has happened over the history of the RDAs, DRIs. But what's important to realize is that you may have abnormal function, abnormal biochemical or biomarker functions in one measure where all of the others are adequate. Depends on how many RDA 1, 2, 3, 4s you want. But if you are meeting this first value, the RDA 1, as it's indicated on this slide, doesn't mean you are meeting all of the benefits of this particular nutrient, depending on which parameter was selected in the first case. So we've already heard that the criteria for recommended allowances may be changing as we look for other benefits other than those in preventing deficiency syndromes. And of course, your curve is going to shift, presumably, to the right as we move from the risk of inadequacy, avoiding deficiency symptoms, to something where we want to avoid the risk of inadequacy for a health benefit, such as the prevention of chronic disease, or any other particular health criteria or functional criteria you want to choose. If we shift this curve further to the right, are we in fact then decreasing that range of adequate or acceptable intake? So it's going to be an interesting communication problem to say, oh yes, eat more of this because it's going to be better for you, but of course you're getting closer to killing yourself at the same time. I wouldn't actually use that as a risk communication tool, but I can see where some might start to think about that. The other is that when we think about these things, we're not necessarily talking about the risks occurring in the same individual. We've already heard a little bit about folic acid, neural tube birth defects, where requirements are set not for the mother, but for consequences for the baby. Dr. Keene talked about vitamin D. There's some very provocative information that inadequate vitamin D intakes may reduce cognitive functioning or learning in children, the offspring of the mother at 2, 3, 4 years of age. As we get better at epigenetic, the science of epigenetics and nutrition, again, we can look at how we might shift this curve, but what are the consequences then when we talk about how do we assess harm in the face of what could be very substantial benefit. So this used to be, for me, the classic toxicological approach. What we want to do is minimize these harmful, dangerous substances, being exposed to them either from the environment or our food. And so for genotoxic carcinogens, we have these kinds of models. The one-hit model, it's just a nice linear extrapolation back to a zero dose, because a zero dose is clearly what you want for anything that's harmful. You can use a multi-stage approach, and there are other calculations that you can use because our mechanistic insights into cytoprotection against genotoxic carcinogens and other agents is sufficiently insufficient that we don't know really how best to draw these lines. But I am concerned when I hear people trying to talk about things like trans fatty acids and say, oh yeah, that's going to be a one-hit thing. One molecule of trans fatty acid, clearly it's not good for you, and extrapolate out. We heard a whole talk about that. One would think by reading the popular literature that one molecule of fructose, of pure fructose, is going to be very harmful, even though we don't ever experience pure fructose in our diet. So these kinds of models have all sorts of problems, and I think with them partly that they don't reflect either our current nutritional state and how we eat and what we eat, and I don't think they reflect very well the kind of harm that we may experience at lower doses. So the current approach now, which you've heard before about using no-Ls, and if there's not enough data there, we'll go to a low-L. There are certainly other approaches. We've heard about margins of exposure and the development of a biomarker, a benchmark dose. And those are certainly other approaches to do this. The problem here is that the uncertainty factor is sometimes used in an arbitrary fashion, sometimes to meet some expected or anticipated outcome, and sometimes we just use established numbers regardless of what compounds we're looking at. And we've already heard about using an interspecies factor of 10 and then an intersubject factor of 10 to get some maximal acceptable daily intake. We also heard that these approaches are based on the most sensitive animal models. And I sometimes wonder whether, in fact, using the most sensitive model extrapolates well when we're really talking about recommendations for a whole population. Maybe for certain subpopulations, that approach makes sense, but when we start translating these data into risk management and risk communication, it can become problematic. So the National Toxicology Program just a few months ago released its report on epigallocatechin gallate. They tested a green tea extract, which was almost pure EGCG. They conducted 1- and 2-year rodent studies. And based on the signs of toxicity, mostly tumor formation, a lot of nasal cancers in these mice. I'm not quite sure how that relates to human conditions, but nonetheless, they were able to calculate from an extensive number in several different mice and rat models a low L of 300 milligrams per kilogram per body weight per day. They applied the uncertainty factor of 100, and now we know that an adequate dietary intake is going to be 200 and daily intake, 210 milligrams per day. A report a couple years ago came out looking at the apparent effect of green tea extracts on hepatotoxicity. There are really very little information regarding dose-response relationships and the hepatotoxicity from green tea extracts. There are very few case reports that we have for this kind of data, but nonetheless, it is human data. They calculated, based on the lowest dose that they could find for this adverse effect of 10 milligrams per kilogram body weight per day, they didn't have to worry about the interspecies uncertainty factor, a UF of 10, and they said that the ADI is 70 milligrams per day. Good, we need to look at that human data. But in fact, when we look at 90th percentile intakes of the catechins, and particularly epigallocatechin gallate, in the US, you can see among men and women, it's about 200 milligrams per day, 3 times what the suggested amount from these calculations are. And in China, it's 6 to 7 times lower than what people are routinely consuming, and apparently without harm. So I think we have to be very careful about inappropriate applications of the precautionary principle here. Particularly in the face of an awful lot of data, and this is a recent meta-analysis of randomized controlled trials of green tea catechins. Most of these trials were done using a green tea extract, and you can see there are small but statistically significant reductions in systolic and diastolic blood pressure. And from a public health perspective, that's really important. Small changes across the whole population in blood pressure can be quite beneficial. And there were significant moderate effects in lowering LDL cholesterol. So how do we interpret data like this using the standard kind of approaches for low L's and no L's and uncertainty factors in the face of what we know human consumption normally is, and more importantly, in the face of that data correlating quite nicely with a human health benefit. So looking at some of those, a different report, looking at a systematic review and meta-analysis of RCTs, they looked at the dose-response reduction in systolic blood pressure by EGCG, and you can tell again at the suggested ADI based on animal and human data, most people aren't consuming enough of these flavan-3-ols to get the benefit of reduction in systolic blood pressure from where you see benefits from 150 to about 400 to 600 milligrams of a daily dose. If you can see the slide okay, you'll see at about 850 milligrams, the dot is on the bottom, and that says there was an increase in systolic blood pressure in this one clinical trial with this one high dose. Oh, this is kind of nice. It suggests to me that there is a lower observed adverse effect level here, a low L, and maybe we just want to stay beneath 850 milligrams a day, or maybe below 601 milligrams a day. I think we can look at this kind of data, which is accumulating for lots of compounds. There have been a number of clinical trials with lycopene and lutein, with coenzyme Q10, where we can generate this kind of data from human data, where you have apparent safe effects. So although this hasn't been adopted by any regulatory group that I'm aware of, an observed safe level or a highest observed intake in human studies where the uncertainty factor is 1 has been suggested where there is insufficient data for a no L or a low L. And this would be nice to have because we actually have a number of compounds, including essential nutrients, where there is insufficient evidence to find an adverse effect. But it would be nice to have some kind of regulatory guidance for doses that just you don't want to go beyond. And so this idea of taking data that exists from human studies and translating it to an OSL or a HOI is certainly an approach that we might consider. The UK vitamin and mineral expert group did a similar kind of calculation for vitamin B12. Nobody's been able to find a harmful effect of vitamin B12, but based on observational and trial evidence like this, they said 2,000 micrograms a day is the highest observed safe level or intake. And again, I think that's useful for informing guidance on safe intakes. So I said I think we need to put all of this in some sort of framework with benefit assessment as well as risk assessment. And now I've learned that toxicologists roll their eyes when they see these 4-step framework for risk assessment of hazard identification, characterization, exposure assessment, and risk characterization. But I would tell you, you can do exactly the same kind of approach using a benefit assessment. The European Commission used value instead of benefit, sort of a less laden term about what the outcome is going to be, but you can do exactly the same kind of thing, a value identification and characterization, an exposure assessment, and then a characterization of that benefit, assuming you get to that point. There is one really important difference between these two columns, and that is that the benefit assessment can only reasonably be done in humans. Risk assessment, as you all know, we can use animal models, but not for benefit assessment. The suggestion has been made that these kinds of benefit risk assessment paradigms can run in parallel, but in fact, you don't begin to integrate until you get to your final characterization of the benefits or risks. And that's something to think about. I would argue that it may be possible if you come to early in this process to identify really significant hazards. You might be able to stop the process early, and conversely, very significant benefits, absent indications of harm, you could do. So one other way of looking at this idea of risk-benefit is to somewhat redraw that classic DRI curve and to understand, as I think there's plenty of evidence to do, that in fact there may be a lower level of additional benefit and an upper level of additional benefit within the range between an RDA and a UL, or if there's no RDA, then we don't have that particular cutoff to worry about. And you can have different shapes. Indeed, it's entirely possible, I think Dr. Keene even referred to this, you may have a zone of overlap where the benefit comes with some risk associated with it. But if the benefit is certainly great enough, you might want to consider it. I've got my heads up for time. There are other more integrated and composite approaches, health-adjusted life years, that have been suggested to use these kinds of summary measures of population health, combining morbidity and mortality data to represent population health in a single number. I'm going to go really quickly here. So these are quality-adjusted life years where you can calculate the area under the curve using quality weights, which are theoretical constructs, and then looking at age of onset and age of death versus life expectancy. When you do these kinds of integrated benefit and risk analyses, there are tons of data, but if you can do it in a tiered approach, you can stop short of going all the way through, as I just suggested. I want to stress that the evaluation of evidence for benefit and risk is asymmetrical. For safety assessment, you certainly want to look all the way from convincing evidence through possible, probable, possible, and insufficient. Where are you on that risk assessment? You stop far short of that for value added benefits after probable. You want something above probable, and it becomes an interesting concept for risk managers. If I have a probable benefit and possible harm, possible risk, how do I begin to weigh that? So I just want to close with a few quick comments. We've seen other Venn diagrams like this one, and I just wanted to tell you that while we've had, I think, a very informative discussion today, I would argue when it comes to risk communication, we seem to be losing in a significant way the war. There are lots of factors that aren't considered when we talk about influencing public perception of risk that don't come in classic risk assessments. If the risk is controllable, possibility of catastrophic or irreversible harm, the novelty of the risk form, like nanotechnology, the extent to which the risk is voluntary, and about whether future generations could be affected. But when I look at communications like this, suggesting there's no way you should follow the dietary guidelines with lots of whole grains because gluten is a biohazard, gluten is a poison, you better not eat wheat, barley, rye, spelt, but if you eat a lot of amaranth, for example, excuse me, and quinoa, maybe you can get by. GMOs, it's a whole other topic, excuse me. But we have safety assessment, we have animal models, we have 20 years of human experience, at least with some GMOs, but this is the public perception, and consumers are voting because they have no idea what they are talking about. It's a Luddite approach that anything that's novel, because they also don't understand the benefits, and there has been terrible communication about the benefits associated with genetically engineered foods. How about this one? You all know this one. If you look for processed ingredients in a food, it's a warning label. We should be eating mostly, if you can read this, mostly raw foods. If you have to, you can cook your foods, but that's about it. Anything else is dangerous. Raw milk, pasteurization, let's just ignore what we know about safety. And irradiation, we all know that makes you glow in the dark if you eat irradiated foods. No concept of weighing the benefits versus the risks. So I just have a closing comment that concerns me. We know about what INDs are about and the FDA's role here. Their primary objective in reviewing INDs for drugs in all phases of investigation is to assure safety. And even in phase 2 and phase 3 studies, they should be conducted so as to permit an evaluation of the drug's effectiveness and safety. But last fall, there was proposed FDA guidance for IND applications, basically applying drug-like criteria for INDs for any food or dietary supplement or food ingredient that would prevent damage to an organ, part, structure, or system of the body. So if I want to determine whether a popular fruit juice has some benefit, for example, in prehypertension or hypertension, I need to file an IND. I'm not quite sure how I'm going to be funded to do that or where I'm going to get the support to do that. It will, of course, delay my starting the clinical trial if I can get to that point. But the problem from what we've heard today is that where are we getting a lot of this valuable information about benefit, about dose response? We're getting them from clinical trials, from foods and from nutrients and observational studies without ever requiring an IND be required for doing that. If we go forth with this, I think the amount of research you'll see, at least in the United States, of these important trials will diminish as people go to other countries and try to get it done. So Paracelsus may be the father of modern toxicology. All substances are poisons. There is none which is not a poison. The right dose differentiates a poison. But we're really not talking about poisons. We're talking about food. We're talking about nutrients. We're talking about food components where there are not only safe doses, there are beneficial doses. And so I'll move from Paracelsus' age and tell you I like Shakespeare better. He says, out of this nettle, danger, we pick this flower, safety. Now, I would tell you the size of the nettle and the size of the flower is going to vary when we talk about nutrition, depending on age, perhaps on sex, perhaps on nutrigenomics. But there is a very important flower to be plucked here, and if we apply inappropriate precautionary principles, if we apply too strictly uncertainty factors, if we don't appreciate that the range or the ratio of benefit to harm has to be carefully considered before we make any final decisions and communicate it, then I'll have made my point. Thank you very much.